exciting day for DueNorth. This is our first investor meeting. So in essence, you get to help us make this a better next meeting. So this will be good.
Quick overview of the company and how we think it's specific and different from a lot of other companies. People have more sophisticated, they've heard of a lot of companies. We're doing things a bit different than the vast majority of companies that I've worked with. One of the things I want to start with is for all of us to get onto the same page of what we're talking about, because personalized medicine hardly hit the literature until about 10 years ago, 2000 to 2001. And even now, if you Google it, you find an awful lot of different definitions. So I think personalized medicine, number one, is going to be huge in medical care. Maybe in a century from now, we'll say that the two greatest discoveries in the last century or two was genomics, number one, in the 50s or 60s, and personalized medicine. And we're not on the end edge at all. We're on the very beginning of personalized medicine, just starting it. We said we needed it in 2000, and now there's just a few compounds, things coming out that lead towards personalized medicine. This will go on for 100 years. Biomarkers, the area that we're interested in, is one critical step in personalized medicine. And again, you can find lots of definitions of biomarkers, but biomarkers in its simplest terms is tissue, blood, tumor tissue, and the important part about a biomarker is that there's a certain outcome that's been validated. It's been tested, it's tested in the lab, it's reliable, and it's reproducible, and it tells you something. And sort of trying to just make an analogy, maybe in the simplest things like a temperature. Okay, you take a temperature, 98 is normal, we know that, and 101 gets higher. We've all lived, men, with the prostate-specific antigen as a biomarker. Not a very good one. It's a pretty poor one, but it's been around forever. And that's a biomarker saying that some people with an elevation may get prostate cancer. So that's kind of the definition of biomarkers. And a biomarker basically helps doctors make decisions. It doesn't help you at all. If you come up with something and it doesn't change medical care, it means nothing. So if it changes medical care and is practical, etc., then it may be useful in medicine. So a biomarker can help a doctor choose the right medicine for the right patient, for the right reason, and at the right dose. And that's where we'd like to be quite a long time from now, but we'd like to sort of start here. And if you want to look at it sort of in a schematic, everyone in this room has a different genetic makeup. Even identical twins to some degree, but the genetics are totally different. If someone develops a tumor in this group, that will have a specific genetic makeup. And if it's a woman and it's breast cancer, it may be very different from this woman with a breast cancer. So there's heterogeneity all over the place. Okay? So again, just personalized medicine, we're talking about biomarkers, but if we were developing a drug, we might go to the lab and develop a drug and say, that's fantastic, there's thousands of them, but it's toxic, you can't take it. You'll die of heart disease, so that drug's out. You have another drug, it's not toxic at all, it's harmless, doesn't cause any side effects, doesn't really benefit you. And of course, if you've invested in the biotech industry at all, you understand that there's an awful lot of drugs who are safe and they fail the phase 3 studies, so they fit into this category. Great drug, theoretically worked, doesn't work. Doesn't prove out in the phase 3 trials. Drug here, not toxic and not beneficial, neither one. So this drug is no good at all. What we're looking for out of this group of people that all look the same, there's no differentiating factor, is that the drug is not toxic and beneficial. So from a drug standpoint, this is sort of what we're talking about for personalized medicine. And in the perfect world, and I'm talking about evolution of what we're talking about today, we have a woman with breast cancer and we pull her down here, and from biomarker tests and personalized medicine, then we just outline the whole treatment regimen. We're not there yet, but at some point in time, we may very well be able to give tools to the physician to say that this patient needs this and this and this and this order, et cetera, et cetera. That's where we'd like to go. And that's what we're attempting to do. our business approach, and this is how I'd like to say that our company is somewhat different. And the next 2 slides will be just the crucial slides for what we're trying to discuss. So what DueNorth has done, we started off, we went through 6 months of various things, met people, went here, went to California, going to merge with another company, etc. We began to look at what's going on. We found out what our real skill set is, both from Charlene and I. I mean, some of it's time, but I've been from the patient care area, drug development, clinical research, etc., marketing, Charlene with patient care. So we really have a spectrum of skills here, very broad. We may not be super experts in everything, but we've done a little bit of all of this. So we think we're very good at picking early research and seeing whether that research can ever make it out to the marketplace. Now, in the biotech world, research world is 5,000, 10,000, there's 5,000 biotech companies, all of the product. There is 50,000, 100,000 research projects within academia. So we chose to not have a laboratory, not to develop drugs ourselves. We go out and look for an unmet need that we think we can solve. Lots of unmet needs in medicine, we list 200 in this room. But what's an unmet need that's really important? There may be research somewhere that solves that unmet need. It really impacts a lot of patients, and it goes through all the pathways we talked about. If the clinical research costs $50 million, we're not going to tackle it. Maybe it takes 10,000 patients, that's off the market. Maybe there's no need for it once it's done because there's a good competitor product that's cheaper. So you fill out the whole thing and you say, this looks like this might really work. Nothing's 100%, we all know that, but really a high probability of getting through these various steps. So we do very, very rigorous due diligence, and there's the time factor of the products that are available out in the community, I mean the research community. The NRC, biological companies, other biotech companies could be a partner, the academia, and there are hundreds. And we sort through those, and we look for solutions that fit like the solution we're going to present today. And this one complex slide we'll walk through because it's pretty important, but the key factor is if I were developing just a biomarker, and I'm saying I'm starting a new company with a fascinating idea, I've been PhD, I've been working on this for 10 years, and it is tremendous. And I need a lab, I need 10 workers, I need a lot of equipment, I need a building, and I'm going to start working on it and develop it. Then this is the typical company back here. I consulted with a lot of these companies, and that's the way the company looks like. And in 3 years, I may get to the standpoint, I may not, and it probably will cost me 15 to 17 million to get there if I'm careful with my funds. So what we've done is say, okay, we've identified an unmet need. We look at the NRC, McGill, other places, universities in the US, and say, this work has been done for this unmet need. We're going to pick it up right here. Well over, this isn't even a time scale, so we can almost put it over here. The work has been done, the basic work looks good. What do we need to do next? Well, we need to do validation in the laboratory, because it has to be translated into a practical test, and then clinical research, and the regulatory approval for a biomarker, which is one of the reasons we've chosen this, is relatively simple. We can do a biomarker and get CLIA lab approval, which is a specific approval designation that does not require Healthcare Canada or FDA approval. We'd like to get that, but it's not required. We can market and sell that drug forever as long as we do the validation steps and everything in that one specific laboratory. That's one potential working model. And now we can get into many, many questions as to, do you want your own lab? Do you want to, how do you want to do this from there on? Do you want to market it? We right now think probably the best business model, like Charlene said, we want to make a difference in medicine, which is why we formed the company. You can't make a difference in medicine if you don't make any money. So that's pretty obvious. So we want a highly profitable company. We think maybe the best profit is probably going to come from developing this biomarker, ready to go, and then joining, working now with Millennium Pharmaceutical companies with 80 or 88 offices worldwide, or Novartis. They will obviously want a percentage of what we make, but they do all the marketing and sales, etc. But that's a later business decision. But those are the kind of things that we might think up on this end. So our first products to market, and I do want to say first, this is a concept, not a product. I know we're investing here, we're thinking about this product. I promise my role right now will be, what's the next product? Because this is just the first two. This has been developed, Charlene's going to take it into operations, and I'm looking for the next product. So This product is breast cancer. And unfortunately, the numbers, and we all know friends and people with breast cancer, 1 in 8 women will be diagnosed with breast cancer in their lifetime. Big, big number. 1 in 2 people will be diagnosed with cancer. Now, the major drug used for breast cancer is Taxol, taxane, started by Bristol Myers Squibb, marketed under various names. Still the best, most used drug in oncology today for all kinds of cancer. Therein lies some other potential biomarker research in the future. It's done the way all drugs now are done. You take this group of people, or this heterogeneous group that you saw, and they give them, all the breast cancer patients get taxane, and some get better and some don't. That's how drugs have been developed in my career over the last 30 plus years. That's how we did it, clinical research. Breast cancer, come in, this group gets taxane, this group doesn't. The taxane group got better, taxane's approved. Now, this is the very first step, to me, in personalized medicine. There are other biomarkers out there. They all say good prognosis, poor prognosis. That's what they're saying. They're very successful, by the way, and Charlene will allude to those. But it gives you the information. This patient has a poor prognosis, maybe they need chemotherapy. I think this is I was just at the ASCO, American Cancer Society meeting, so there's other biomarkers coming in that category. Prostate, good prognosis, poor prognosis. This is the very first personalized medicine biomarker that I'm aware of. No, sorry, HER2/neu, been around a while. That's personalized biomarker, so not the first. But the first one that tells you a large group of patients given a breast cancer and analyzed with this biomarker, you may respond to a taxane, you have a very high likelihood, or you may not respond to a taxane. Does not mean that the cancer specialist has to use that data, but it adds to the other information that he has. From a breast cancer patient, if you don't think you're going to respond to a taxane, this first key drug, why would you want to get the side effects, get the drug for 6 or 8 months to find out it doesn't work, to go on to, quote, second-line therapy? That's the simple argument. It can get very complex, as you know, and everything else, but this will tell you whether one responds to taxanes. And my final closing comments are, this is just the beginning of what we consider our biomarker program. We may well continue to work with NRCs to develop another biomarkers along those lines. Obvious questions come up in the taxane response. What about the secondary drug, adriamycin? Does that drug work in breast cancer? So we can continue to work there. And the 2 final areas that we are very interested in is ovarian cancer, diagnosing it at a treatable stage, which is very hard. About 80% of the patients with ovarian cancer are diagnosed in advanced disease, uncurable almost. We are starting to look, just doing our due diligence and talking to experts in the field on ovarian cancer. And the final one that Dr. Varma, myself, were very interested in is sarcoma, a disease that hits a lot of people, hits a lot of young children. A good friend lost his leg and then lost his life to it. And are there various biomarkers, which there may be in the sarcoma field, that we can utilize for diagnosis for